This is Leishmania. The brief review of what it is: it's a unicellular eukaryotic parasite and an obligate intracellular protozoa, which just means that it can't live without a host organism. Common hosts include humans, bats, dogs, rodents, possums, anteaters, sloths, and marsupials, with dogs being the main reservoirs for the parasite. The supergroup is excavata, and the subgroup is euglenozoan. The life cycle is that it starts as a promastigote within a female sandfly's gut, and then the sandfly goes to bite a human or another organism. The promastigotes are released into the bloodstream, during which it encounters a vast variety of cells, but it typically goes after the macrophages in the bloodstream. This is where the development of the promastigote to the amastigote occurs. This infection then spreads to other cells in the body and is then picked up by other female sandflies whenever they bite the human, and then the cycle begins anew because the amastigotes return to their promastigote form within the gut once again. The Leishmania parasite is able to survive this environment through various adaptations, the first of which is its inhibition of apoptosis, which is a mechanism that induces cell death. This can be used to prevent various immune responses by preventing the spread of the parasite, as well as viruses. And by inhibiting this allows for the further spread and increase in severity of the infection. A similar process to this is pyroptosis, which causes rapid cell lysis and elicits an immune response. It can inhibit this, which allows immune evasion and further development of the parasite.
So Leishmania are flagellated protozoan parasites with, like we said, a two-stage life cycle. Inside the mammalian host, they exist as amastigotes, which are shown on this bottom picture, where they are small, round, and non-motile forms, and when a sandfly takes a blood meal, it's going to ingest these amastigotes, which will then transform midgut into promastigotes, which I have shown on this top picture, and that's the elongated, flagellated form adapted for survival and multiplication in the insect. So both forms are highly dynamic in their plasma membrane, and it's made of phospholipids. The membrane is rich in ergosterol instead of cholesterol. This membrane is studded with molecules anchored by glycophosphodiesenol, which is for short GPI, which help the parasite interact with its environment. And beneath it, there's a network of microtubules plus a specialized flagellar pocket to support cell shape and direct secretion and uptake. And you can see this black asterisk in here is showing the flagellar pocket, and the yellow arrow right here is showing the microtubules forming within the flagellum. So the biggest difference between the forms is their surface coats. The promastigotes in the sandfly have a thick layer of lipophosphodiesenol, which for short is LPG, and GPI anchored proteins, and then after being taken up by the host macrophages, the parasite converts into the amastigote form, and the LPG coat is greatly reduced. The membrane becomes richer in simpler GPI lipids, GPILs for short, and transport systems adjust to low pH and oxidative stress, and these changes allow the parasite to replicate inside the phagolysome. So on this top picture here, you can see that this dense dark line actually is the plasma membrane, and then the light dense outer layer is the glycocalyx, and in promastigotes this is very thick with the lipophosphodiesenol and the GPI anchored proteins that protect the parasite, while beneath it lies the cytoskeleton, all of that. And then on this bottom picture shows the amastigote form, and you can see that there still is a plasma membrane and GPI linked proteins. That's those little dots right there. But the prominent lipophosphodiesenol and the dense glycocalyx from the promastigote stage is very largely absent in the amastigote, giving the surface a much simpler appearance.
Leishmania parasites face many stressors, both environmental and physiological. Environmental conditions such as temperature, pH, and nutrient availability play a critical role in regulating parasite survival and differentiation. As Leishmania transitions between the sandfly vector and the mammalian host, it encounters new pressures like oxidative stress, immune response, and drug exposure. These stressors push the parasite to adapt, and over time, they can select a more resistant strain. One way Leishmania adapts is through the change in its genome. Research shows that parasites can alter both chromosome and gene copy number. For short-term adaptation, chromosome amplification plays a big role, while long-term adaptation often involves a change in the gene copy number. Interestingly, these genetic shifts are reproducible but unique to each strain, meaning Leishmania evolves in a way that's tailored to its specific environment. Adding to this complexity, Leishmania's genome is highly plastic. Across different isolates, scientists have observed single nucleotide polymorphisms, structural variations, and even whole chromosome changes. This plasticity contributes to the drug resistance, making current treatments less effective and the search for more new and effective drugs more challenging. Altogether, environmental stressors and genome plasticity work hard and hand in hand to drive Leishmania's adaptability. While this helps the parasite survive under hostile conditions, it also makes controlling the disease and developing effective treatments a constant challenge.
The three main types of diseases that this parasite causes is cutaneous leishmaniasis, which causes skin lesions, mucocutaneous leishmaniasis, which causes destructive lesions on the nose, mouth, and throat, and visceral leishmaniasis, which is the most severe and it targets the internal organs.
Cutaneous leishmaniasis is a parasitic disease often referred to as the great imitator because it mimics a wide range of dermatosis. Cutaneous leishmaniasis, shortened by many physicians to CL, is the most common form of leishmaniasis, with an estimated 600,000 to 1 million new cases each year. CL is typically found in people who suffer from poverty, malnutrition, and poor housing conditions. Regions experiencing war-related migration, such as Syria, Turkey, and Jordan, report particularly high number of cases. CL causes lesions and ulcers to exposed body parts. These lesions vary in appearance, which can often lead to misdiagnosis. While the condition causes permanent scarring, it is generally not life-threatening. Mucocutaneous leishmaniasis, shortened to MCL, is less common than cutaneous leishmaniasis, but significantly more severe. It is endemic to Central and South America, where environmental conditions favor transmission. MCL develops after cutaneous leishmaniasis is untreated or inadequately treated, and it occurs more frequently in individuals with weakened immune systems. The disease is caused by Leishmania brasiliensis and results from parasite dissemination to mucosal tissues, such as the oral, nasal, and pharyngeal cavities. This process often leads to extensive tissue destruction and disfigurement. While MCL is rarely fatal, it causes substantial morbidity. Fortunately, it can be treated with pentavalent antimonials.
This is visceral leishmaniasis, and it is one of the most neglected tropical diseases worldwide, with the highest number of cases found in the Americas, East and North Africa, and West and Southeast Asia, with 50,000 to 90,000 new cases reported worldwide each year. Clinical symptoms often don't develop weeks to months after infection has occurred. Common symptoms for diagnosis include prolonged fever, significant weight loss, an enlarged liver and spleen, which is hepatosplenomegaly, and pancytopenia, which is the decrease in white and red blood cells and platelets. The drop in blood cells counts can lead to fatigue, higher chances of getting infections, and easy bruising or bleeding. People at high risk of exposure to this parasite include children, individuals living in rural or high poverty areas, and those with weakened immune systems, especially those with HIV. Patients with HIV can make the diagnosis of BL difficult, which leads to higher rates of failure in treatment plans. Diagnosis can be done with PCR testing of blood samples or by directly detecting the parasite in bone marrow aspirates. This diagram is a comparison of all three main types, and then these are our works cited.